Welcome back to our channel where we explore fascinating topics in medicine. Today, we're diving into the intriguing history and characteristics of Corynebacterium, specifically focusing on the infamous Corynebacterium diphtheriae, the bacterium responsible for causing diphtheria. Let's get started. Our story begins in 1884 when German bacteriologists Edwin Klebs and Friedrich Loeffler made a groundbreaking discovery. They identified a bacterium that was later named Corynebacterium, also known as the Klebs-Loeffler bacillus. This bacterium was found to have a club-shaped rod morphology, belonging to the group of gram-positive, aerobic bacteria. Corynebacterium is characterized by its non-motile nature and a unique cellular arrangement. When these bacteria divide, they exhibit a snapping division, resulting in groups of angular and palisade-shaped cells. This distinctive arrangement has been likened to the appearance of Chinese letters. In addition to their cellular morphology, Corynebacteria are known for the presence of metachromatic granules, also called Babes-Ernst bodies. Non-pathogenic diphtheria lacks them. These polyphosphate inclusions give the bacteria a barred or striped appearance. Corynebacteria can be found in various habitats including soil, water, plants, and even certain food products. Corynebacterium also has a significant presence in the human body. They can be found in the skin, upper respiratory tract, gastrointestinal tract and urogenital tract. However, our focus today is on one particular species of Corynebacterium, Corynebacterium diphtheriae, the causative agent of diphtheria. Diphtheria is a severe respiratory infection caused by Corynebacterium diphtheriae. It primarily affects two types. 1. Nasopharyngeal diphtheria, which involves the pharynx and larynx, and 2. Cutaneous diphtheria, which affects the skin. The incubation period for diphtheria is typically 2 to 5 days. The initial symptoms of diphtheria are often mild and include a sore throat, malaise, and low-grade fever. However, the hallmark sign of diphtheria is the development of a grayish-white pseudomembrane on the pharynx, larynx, or tonsils. Now, let's dive into the pathogenesis of diphtheria. Corynebacterium diphtheriae is transmitted through respiratory droplets, nasopharyngeal secretions and sometimes contaminated objects. Upon entering the body, the bacteria adhere to mucosal epithelial cells. Once the bacteria have established themselves, they produce a potent exotoxin that causes localized inflammation, tissue destruction, and necrosis. This toxin can also spread through the lymphatic and hematological systems, affecting organs such as the myocardium, kidneys, and nervous system, result in myocarditis, neuritis and nephritis. The virulence factors of Corynebacterium diphtheriae contribute to the severity of the disease. 1. Diphtheria toxin, which blocks protein synthesis. 2. Dermonecrotic toxin, which is a sphingomyelinase which increases vascular permeability. 3. Hemolysin. 4. Cord factor known as toxic trehalose. The most well-known factor is the diphtheria toxin itself, which is an exotoxin. Interestingly, the gene for this toxin is encoded by a bacteriophage, a virus that has inserted itself into the bacterium's genome. The formation of a pseudomembrane is a characteristic feature of respiratory diphtheria. This pseudomembrane is not a true membrane but a deposit of proteins, live and dead cells, that covers various parts of the respiratory system, including the tonsils, uvula, palate, nasopharynx, and larynx. Host. When it comes to diagnosing diphtheria, there are several methods available. Throat swabs, pieces of the membrane, or biopsy tissue can be used for direct microscopy and culture on blood tellurite agar. which shows characteristic black colonies due to tellurite reduction by Corynebacteria. Toxogenicity testing, such as the ELIC test or animal inoculation tests, can be performed to confirm the presence of toxin. Other diagnostic methods include PCR, serum antibody testing using ELISA, and tissue culture neutralization assays. Prompt treatment is essential for diphtheria. Toxin-neutralizing antibodies, known as diphtheria antitoxin, are administered along with antibiotics such as penicillin or erythromycin to stop toxin production. Strict isolation measures are crucial to prevent further transmission. Prevention plays a vital role in combating diphtheria. Vaccination with a trivalent vaccine containing diphtheria toxin, pertussis vaccine, and tetanus toxoid, DPT or DTP vaccine, is the primary method to protect against this disease. And there you have it. We've explored the captivating world of Corynebacterium diphtheriae and its association with diphtheria. From its fascinating history to the diagnostic methods and treatment options available, we hope you've gained valuable insights into this bacterial pathogen. As always, stay curious and keep exploring the wonders of microbiology. Thanks for watching. Don't forget to subscribe.